All right, today we are putting on our detective hats. We're diving into a medical mystery where the crime scene is the human bloodstream. The clues are literally microscopic, and the stakes, well, they couldn't be higher. You know, think about it. Most of us have had blood drawn at some point. It feels so routine, right? But what if that one little vial, that single drop of blood, could actually whisper some of the deepest secrets of our health?
Well, it absolutely can. And the hero of our story today is a test you've probably heard of, maybe even had dozens of times, the Complete Blood Count, or CBC. It seems so simple, but we're going to see how this one report can become a detective's most powerful tool for hunting down cancer. So our first case file opens with a really troubling disappearance. We're going to investigate what happens when the cells that are absolutely essential to keeping us alive just seem to vanish. Okay, meet our patient. He's a 68-year-old guy, and he's just not feeling right. He's tired all the time, bruising really easily. Now take a look at his CBC results. His hemoglobin, which is all about his oxygen-carrying red blood cells, is way down at 7.8. That totally explains the fatigue. His white blood cells, his immune system's army, down to 2,800. And his platelets, the little guys that stop bleeding, they've crashed to 40,000. When all three of these are low like this, we have a name for it: pancytopenia. So, this leads us to the single most important question a medical detective can ask here. If all the good, normal, hardworking cells are gone, what on earth has moved in and taken over? And here's our answer: acute leukemia. The best way to think about it is this: your bone marrow is like a super busy factory pumping out all the blood cells you need. But in acute leukemia, that factory gets completely overrun by these useless, malignant squatter cells called blasts. They don't do any work, they just multiply like crazy and literally crowd out the production of every healthy cell you need to survive. This brings us to our first huge detective's clue, a real pearl in medicine. If you see pancytopenia, meaning all the counts are low, and you see those nasty blast cells in a blood smear, you have to assume it's leukemia. It becomes your #1 suspect until you can prove it's something else. Now here's a fascinating twist. The very treatment for cancer, chemotherapy, can actually create a really similar situation. See, chemo is designed to kill any cell that divides rapidly, which is exactly what cancer does. The problem? The healthy cells in our bone marrow are also some of the fastest dividing cells in our entire body. So the chemo hits them too, basically shutting down the factory and causing all those cell counts to plummet. And this is exactly why doctors watch that CBC like a hawk after chemotherapy. They're especially watching a type of white blood cell called a neutrophil, our frontline soldiers against bacteria. If that count, the ANC, drops below 500, a simple infection can become life-threatening. That number, 500, is a critical red line. Okay, time for our next case. We've just seen what happens when there are too few cells, but what happens when the factory goes haywire in the opposite direction and starts producing way, way too many? Take a look at this number, just 180,000. With no context, it's just a big number. But on a CBC report, this is a 5-alarm fire. So this was the white blood cell count for a 45-year-old man. Remember, the normal range usually stops around 11,000. So this isn't just high, it's an absolute flood of white blood cells. But here's the kicker. The real clue isn't just the sheer number. When you look under the microscope, you see a huge increase in one very specific kind of white cell, a basophil. And the diagnosis? Chronic myeloid leukemia, or CML. What's happening here is that a tiny genetic mistake basically flips a switch in the bone marrow factory and locks it in the on position. It just churns out white blood cells nonstop. Now, the big difference from the acute leukemia we saw before is that with CML, these extra cells are actually more mature and can function for a while, which is why it's called chronic. And that leads us to our second big detective's pearl. If you see a CBC with a white count that is through the roof, and specifically you see a lot of those basophils, the prime suspect is CML. It's a classic, almost unmistakable signature. All right, so far, our investigation has been all about the numbers game, too high or too low. But sometimes the most revealing clues aren't in the numbers at all. They're hiding in plain sight, in the actual shape and appearance of the cells themselves. Okay, so picture this. We have a 65-year-old patient who feels totally fine, comes in for routine checkup. The CBC comes back with a high count of lymphocytes, which are a type of white blood cell. But here's where it gets weird. When the lab tech goes to make the glass slide to look at the cells, a bunch of them are so fragile that they literally get smushed in the process. They look like little smudges on the glass. And these smudge cells are the smoking gun. They are a classic, textbook sign for chronic lymphocytic leukemia, or CLL. It's because the cancerous lymphocytes in CLL are just not as tough as healthy ones. It's a fantastic visual clue that tells you exactly what you're dealing with. Let's look at one more. We have a patient who is already fighting breast cancer that has spread. She develops anemia, meaning her red blood cell count is low. But when we look at her blood smear, we see something incredible. Her red blood cells, which are supposed to be nice and round like little donuts, are stretched out into the shape of teardrops.
This shape tells such a vivid story. It's telling us that the bone marrow factory is under siege. It's being physically invaded and squeezed, in this case by the breast cancer cells. As the healthy red blood cells try to escape this overcrowded, damaged factory, they get physically deformed and stretched into this teardrop shape. It's a sign of a physical invasion.
So, we've investigated cases of too few cells, too many cells, and cells that are just plain weird looking. After all this detective work, what have we really learned? What are the big takeaways?
Well, it really boils down to a pretty simple rule book. First, context is king. You always have to look at the patient, not just the numbers. Second, extreme numbers, super high or super low, are always a massive red flag that you have to investigate. Third, don't ever forget that the shape of a cell can tell you a story all on its own. And finally, and this is the most important part, it's all about pattern recognition. Putting all these clues together is how you crack the case.
You know, the complete blood count is just one test. It's just one chapter in the incredible storybook of our own biology. And now that you've seen the kinds of life and death stories that can be hidden in a single drop of blood, it just makes you wonder, right? What other secrets are just sitting there, waiting for us to learn how to read them?